Welcome to our presentation on psilocybin and the human brain. Today we're going to be talking a lot about psilocybin, which is a psychedelic compound found in a species of mushrooms called magic mushrooms, and really how it relates to organic chemistry. Over this past year, we have learned so much about organic chemistry and have this opportunity to see how it plays out in real life. To go into our abstract, like I said, it's a naturally occurring psychedelic compound, but it acts on serotonin receptors. We call psilocybin a prodrug because it is activated after administration. It turns into psilocin almost immediately upon ingestion through dephosphorylation, and we'll get into that a little bit later. But recent preliminary studies have shown that it has a lot of potential for treatment of alcoholism, addictions, depression, anxiety, and even OCD. So we're going to be looking at both the inactive form of the drug psilocybin and the active form of psilocin. For introduction, we have a picture of psilocybin, the structure of it, and then what it looks like as a mushroom. It really gained popularity leading up to the 70s, and then in the 70s, it became a Schedule I drug, which means that there's high abuse risk, no deemed medical use, and also illegal. So after becoming a Schedule I drug, it lost a lot of steam. But in recent years, there's been a renewed interest in this magic mushroom because it has shown a lot of potential for neuroscience breakthroughs and therapeutic effects. So scientists are really getting excited about what this could mean for future treatments of anxiety and depression, and also breaking and overcoming difficult addictions.
Right, I will be going over the history of psilocybin in organic chemistry. Psilocybin entered organic chemistry through something called ethnobotany, and its culture is typically based in Mesoamerica, as they used psilocybin-containing mushrooms for centuries. However, Western science only became aware of them after Wasson's 1957 report. In 1958, Albert Hofmann isolated and synthesized psilocybin, which made it a key moment where a naturally occurring compound became a defined organic molecule, and this work established psilocybin as part of a broader class of tryptamines and positioned it in an area of synthetic organic chemistry that it had not formerly been known in.
Following its synthesis, psilocybin became a major focus of research in the 1960s, particularly in psychiatry and medicinal chemistry. Also, the hippies out in the world were researching this drug as well. However, since the hippies were using this drug so much, legal restrictions in the late 1960s halted most research, effectively pausing progress in understanding its structure and activity relationships. Over the past 2 decades, there has been a resurgence, which has often been called the psychedelic renaissance, where psilocybin is again studied as an organic molecule with therapeutic potential. Modern research revisits its chemical structure, analog development, and its role as a model compound for understanding how small structural changes influence brain function. Psilocybin's history reflects the trajectory of organic chemistry itself, from natural product isolation, synthetic control, to modern structure-driven drug development. So how does this work? After you ingest psilocybin, it's converted to its active form, psilocin, because psilocybin on its own doesn't bind to any receptors. It has to be dephosphorylated. This is catalyzed by enzymes such as alkaline phosphatase in the intestines and liver. Psilocin is then absorbed into the bloodstream and readily crosses the blood-brain barrier. Once in the brain, psilocin binds to serotonin receptors on neurons as a partial agonist, activating G protein signaling pathways and altering normal communication between cells. So the starting molecule of this pathway is glucose 6-phosphate, which is produced by glycolysis, which is something that most living organisms do in their metabolic process. The glucose 6-phosphate is synthesized into erythrose 4-phosphate, or E4P, using the pentose phosphate pathway catalyzed by transilidose. The E4P is combined with phosphenoenol pyruvate, or PEP, which is found and derived from glycolysis, in the Shikimate pathway to produce tryptophan. After tryptophan, we are gonna see what this molecule does.
The molecule of glucose has a weak nucleophile on the OH group of the 5th carbon. To add the phosphate group, we need to change the stability of the oxygen to make the substitute attack on the same oxygen. By changing the oxygen into a strong nucleophile, which will allow oxygen to attack a positive species, making an SN2 reaction that we've seen many times before. First, we need a base in form of an enzyme called hexokinase, a group that transfers a phosphate group from one molecule to another. After the phosphate group is substituted, glucose 6-phosphate is formed. After glycolysis, glucose 6-phosphate is oxidized to form 6-phosphogluconolactone, a cyclic ester. NADP plus an electron carrier is also produced. This is essential for the rest of the reaction. This is a rate-determining step for the rest of the reaction. Next, a carbon is cut off and CO2 is released. Well, the electrons released from this reduce NADP plus to NADPH. This 5-carbon molecule is called ribulose 5-phosphate. In this phase, ribulose 5-phosphate goes through a structural change, so the carbonyl group inside the chain is moved to the end, resulting in ribose 5-phosphate. Xylulose 5-phosphate comes from this step. It acts as a carbon donor and forms cetoheptulose 7-phosphate. The fragmented 3-carbon dihydroxyacetone unit becomes glyceraldehyde 3-phosphate. After the enzyme transaldolase cleaves off the 3 carbons, the remaining 4 carbons becomes the essential reagent that reacts with phosphophenol pyruvate. Corsmate contains a reactive ring structure that can be modified to form tryptophan. First, an enzyme called anthralite synthase uses glutamine as a nitrogen donor, allowing the nitrogen group to attack corsmate and release pyruvate, forming anthranilate. Anthranilate then reacts with PRPP, a molecule containing an activated ribose phosphate group. The nitrogen on anthranilate acts as a nucleophile and attacks the activated carbon on PRPP, forming phosphorbosyl anthralite through a substitution reaction. The molecule then undergoes several rearrangement and remodifications that stabilize the structure and help form indole. Finally, indole reacts with serine in the presence of tryptophan synthase, forming a new carbon-carbon bond and producing tryptophan as the final product.
The part of the mechanism which I will be going over is decarboxylation, in which a carboxyl group is removed as carbon dioxide, and the goal is to generally create a more stable and reactive intermediate. So within psilocybin, the synthesis occurs when tryptophan undergoes enzyme-catalyzed decarboxylation to form tryptamine, and it's crucial because it creates the amine backbone that allows for future transformations, including hydroxylation and methylation, additionally phosphorylation. So the reaction is driven by the release of stable CO2 and the formation of resonance-stabilized intermediates, which are usually assisted by phosphate. Without the decarboxylation step, the molecule would remain too polar and unreactive to proceed, so decarboxylation is essential for building psilocybin.
In the second step, tryptamine undergoes aromatic hydroxylation, in which enzyme cytochrome P450 essentially adds a hydroxyl group to the 4 position of the benzene ring of tryptamine. This is done through the use of the iron oxo complex on P450. Normally, the iron oxo would react through a radical mechanism which takes a hydrogen atom with its electron, though in this mechanism it behaves more like an electrophilic aromatic oxidant. The iron oxo complex turns molecular oxygen into an activated oxygen, which adds across the double bond of the benzene, creating an arene oxide. This is debated exactly how the intermediate forms, but the arene oxide is similar to the epoxide reactions we had in the class. Once the arene oxide forms, the ring loses aromaticity, which causes a carbon to become electron deficient, and this creates a cation-like intermediate. Then a hydrogen migrates to the electron-deficient carbon, which breaks the epoxide. This is called an NIH shift, which is named after the researchers at the National Institute of Health which discovered it. The negative oxygen reforms as a ketone on the fourth carbon. Then, a base present in the cell deprotonates the number 5 carbon, causing an acidic keto-enol tautomerism, which restores the aromaticity and gives oxygen a negative charge. The oxygen then attacks an acidic hydrogen, leading to a hydroxyl group on the number 4 carbon and our product 4-hydroxytryptamine.
Diving into the phosphorylation of 4-hydroxytryptamine, first I want to just point out on the left side we have 4-hydroxytryptamine. That's our starting material or our substrate. In the middle we have our enzyme catalyst, 4-hydroxytryptamine kinase, and then ATP that goes into ADP. Our right side is our phosphorylated product, which is the main event. That's what's happening here. And that turns into 4-hydroxy and methyltryptamine. So this reaction adds a phosphate group to the hydroxyl group on the aromatic ring from 4-hydroxytryptamine, and ATP provides the phosphate, and the enzyme 4-hydroxytryptamine kinase catalyzes the transfer. So basically, the enzyme transfers a phosphate from ATP to the oxygen atom, which produces a phosphate ester and ATP, which is shown here in this slide. In this reaction, the hydroxyl group on 4-hydroxytryptamine is being phosphorylated, which means that there's a phosphate group that's being added to the molecule. The oxygen atom in the hydroxyl group has lone pairs of electrons, so it can act as a nucleophile and attack the terminal phosphate group on ATP. As this happens, a new bond forms between the oxygen and the phosphorus atom, and at the same time, the bond that holds the phosphate onto ATP breaks, which then releases ADP. ATP acts as the phosphate donor in this reaction. So the final product, as you can see here, is a phosphorylated molecule that contains a phosphate ester functional group. Finally, moving on to the products. As you can see here on the left, we have our final phosphorylated product, which is a phosphate ester. So the last step of this reaction is showing that the phosphate group has been successfully transferred onto the 4-hydroxytryptamine. So when the new bond forms between the oxygen and the phosphate group, as shown at the top of this slide, the oxygen has a temporary positive charge because it is bonded to too many atoms, and so it loses a hydrogen atom, and the electrons from that bond go back to the oxygen and remove the positive charge, making it more stable. And during that time, ATP loses its terminal phosphate group and becomes ADP, or adenosine diphosphate, also shown on this slide on the bottom right. And ATP acts as the phosphate donor in this reaction. So the curved arrows in this mechanism just show how these electrons are moving, the bond formation, and then the overall transfer of electrons. So this is a phosphorylation reaction where a phosphate group from ATP is added onto the hydroxyl group.
In the last step of the synthesis, 4-hydroxy and methyltryptamine is converted to psilocybin, which is our final product via 2 SN2 methylations catalyzed by the enzyme N-methyltransferase. A required reagent for this reaction is S-adenosyl methionine, also known as SAM, which is converted to S-adenosyl L-homocysteine, or SAH, through this reaction. In the electron pushing mechanism, the lone pair on nitrogen attacks the methyl group present on the sulfur on SAM. This is because the methyl group is attached by a positively charged sulfur, making it an excellent leaving group. Once the methyl group is on the nitrogen, making it positively charged, aspartate, which is found in the cytosol, acts as a base, deprotonating the nitrogen, resulting in a neutral charged secondary amine. This process is repeated with a new SAM, replacing the single hydrogen left on the nitrogen with a methyl group with aspartate deprotonating nitrogen once again. This leaves us with our desired product, psilocybin, as well as 2 SAH, which will be fed into the SAM cycle to be remethylized.
Once psilocybin has entered the digestive system, phosphatase enzymes present in the liver will dephosphorylate the compound. There's not a specific enzyme that does this, so aspartate is used as a placeholder. This is done through the activation of water into a strong nucleophile which attacks the phosphorus atom. This atom is highly electrophilic since the multiple oxygens make it partially positive. Once the pentavalent phosphate intermediate forms, the double bond that was broken is reformed, breaking the bond of the oxygen connected to the benzene ring. The negatively charged oxygen then attacks the hydrogen of nearby water, resulting in a hydroxyl group and our product, psilocin.
Psilocybin is rapidly converted in the body to psilocin, which is the active form that readily crosses the blood-brain barrier. Due to its structural similarity to serotonin, psilocin binds to serotonin 5-HT2A receptors in the brain and acts as a partial agonist. This binding involves multiple non-covalent interactions, including hydrogen bonding, and activates G protein signaling pathways within neurons. As a result, downstream signaling increases, glutamate releases, leading to an enhanced neuronal excitation and altered patterns of brain connectivity. These changes produce the characteristic psychedelic effects, including altering perception, cognition, and a more globally interconnected pattern of brain activity. While the exact mechanism of psilocybin are not yet fully understood, it shows potential for a use in addiction treatment. Mechanistically, psilocybin alters patterns of brain connectivity, which may reduce reliance on entrenched habitual neural pathways, commonly associated with addiction. By disrupting these rigid patterns, it may create a temporary window in which the brain is more adaptable, allowing for the formation of healthier behavioral responses. Although this therapeutic effect is still being actively studied and is not fully established, current evidence suggests it may serve as a useful adjunct to structured treatments such as psychotherapy and lifestyle interventions, rather than a standalone cure.
Psilocybin is an organic compound found in certain mushroom species where it is biosynthesized through enzyme-catalyzed pathways involving well-defined organic reactions. When consumed by humans, psilocybin undergoes dephosphorylation to form psilocin, its active metabolite, which interacts with serotonin receptors in the brain. This interaction leads to altered patterns of neural connectivity and increases global communication between brain regions, producing psychedelic effects. These changes in brain activity may also have therapeutic potential, particularly in the treatment of addiction. By disrupting rigid habitual neural pathways and promoting increased neuroplasticity, psilocybin may help facilitate the formation of healthier, non-addictive behavioral patterns when combined with therapy and lifestyle changes.